जरा मैं रिकॉर्डिंग शुरू कर दूं, फिर शुरू करिए आप। यस स्टार्ट।
जी सर।
यस स्टार्ट।
मैं सर को बता दूं कि स्टार्ट कर रहे हैं ना। जी सर, जरा मैं आपको इंट्रोड्यूस करवाने के लिए निरुपमा मैम से कह देता हूँ, हमारी रिपोर्टर हैं। 2 मिनट आप फोन पे बने रहिएगा सर, ठीक है?
ओके, यस फाइन।
So it's an absolute honor and privilege to have Dr. Shankar Mondal with us today. I welcome Dr. Shankar Mondal on behalf of GPGC Bajpur, SRBS, and on my own personal behalf. I often take advantage of being his junior from Department of Animal Science, Royal University. This lecture is also was a last-minute fix, and I am really very thankful for Dr. Shankar to Dr. Shankar Mondal for accepting my request at the very last minute. He is with us today in spite of his great personal loss, as his father-in-law passed away a few days back. I welcome you, sir, and I now call upon Dr. Nirupama Dalakoti to give a formal introduction of Dr. Shankar Mondal to our audience. Over to you, Dr. Nirupama Dalakoti.
Thank you, sir. Good morning, everyone. I welcome you all in the today's session of Invited Lecture Series on Biological Science 2020. I am honored to introduce our esteemed speaker of the day, Dr. Shankar Mondal, Assistant Professor at LSM Government PG College, Pithoragarh, Uttarakhand. Dr. Mondal is PhD from All India Institute of Medical Science, AIIMS, New Delhi, and has worked on antimicrobial science, AIIMS. And has worked on antimicrobial and immunomodulatory effects of Tulsi. Dr. Mondal has 9 years of teaching experience and has won many accolades in the field of research and won many awards like UK India Science Bridge Fellowship from University of Nottingham, UK, ICMR Research Associate Fellowship and Senior Research Fellowship from Indian Council of Medical Research, New Delhi. AIIMS Institutional PhD Fellowship, CSIR NET Junior and Senior Fellowship and National Talent Research Fellowship. Dr. Mondal's research work has been published in many reputed national and international councils like International Society of Horticultural Science Belgium, Royal Pharmaceutical Society of Great Britain Valley, Blackwell Publications UK, Journal of Pharmacy, Pharmacology UK, etc. Dr. Mondal has also edited 2 books which are published by ACTA Belgium and Science Sanchav New Delhi, and 6 articles having cumulative citations more than 270. His most important research publication is related to cardiology, reducing cardiovascular disease risk factor, and double-blinded randomized control trials. Dr. Mondal has attended more than 3 dozen conferences, seminars, workshops. Dr. Mondal is member of various scientific bodies which includes Indian Immunological Society, Royal Pharmacultural Society of Great Britain, United Kingdom, International Society of Horticultural Science, Belgium, Association of Physiologists and Pharmacologists of India. Sir, we are very privileged to have you on this lecture series. Now, I request Dr. Mondal to deliver his lecture on his topic, Immunology against Coronavirus. Sir, please.
Thank you, ma'am, for such a long introduction that did not require. Anyway, I'm happy to learn these new technologies for this online presentation. So my topic is immunity against the coronavirus. As we are all coping with this lockdown measures because of these simple viruses. So this virus is very simple to say as a corona, but it has many implications. So the topic itself says the glass is half filled because we do not have the whole knowledge about this virus and its transmission and all the immunity involved in. So let's move on to, this is my overview of the lecture, in which I will briefly introduce about the viruses and their replication modes, then come to the coronaviruses and its possible origin. Then I'll be talking about the common symptoms of the coronaviruses. Then I'll be talking about the immunity against the coronaviruses, which mainly will focus on the cytokine system and clearance of the virus, and if there is a possible recurrence, and then is there any way ahead how to manage this virus, because treatment is not 100% available as of now. as we know that this diagram is showing these two human body, and human body is a good source of nutrition. Nutrition for whom? Viruses, bacteria, fungi, and parasites. So they try to enter in our body, but our body is also very smart. How? They try to stop the entry as a form of physical barrier. So we do have only the nasal opening, this gastrointestinal opening, and the ear opening is the only source from which these viruses can enter in our body. And these viruses are very small in size, very tiny. That means they are very small entity. We can even not call them as a organism because they do not have a definite plasma membrane. So it cannot survive its own. And if we see its size, then it's a very small size, and that ranges from 20 to 500 nanometer. And its basic structure is very simple. It has a protein coat and a genetic material. And I'm going to show you a simple structure of a virus. You can see this magenta color nucleic acids. This nucleic acid may be DNA or RNA, and it has a protein coat outside it. And this is a naked virus. Some of the viruses may have a outer covering as well, that is called as envelope. So this is what this envelope. And this envelope contains many kind of the spikes. They can these spikes are very specific for the attachment. So this virus cannot replicate its own. So they have at least 7 steps when they do want to survive, replicate, and their numbers want to increase. So here 7 steps. What are these? First step, that is the entry to the cell. So this square box is representing the cell membrane, and this virus is outside. So this green color entity is a cartoon representative for the virus, and this red color signal is a receptor. So virus, whenever wants to entry from the outside of the entry, it need to bind to that receptor. So once it binds to the receptor, it can come inside through the endocytosis or an lysis of the plasma membrane, and they uncoat themselves. When we say the uncoat, that means the protein covering, it has to be removed. The next step is to expression of the genetic material. Viruses may be of RNA genetic material or DNA material. If the RNA is there, RNA can be of again 2 types: single-stranded RNA, double-stranded RNA. Even if it is single-stranded, it can have a positive strand or negative strand. Positive strands means what? It can directly replicate themselves by utilizing the replicatory mechanism and producing the proteins directly. And when this viral genome attaches with the main DNA, it starts their own replication. Then late gene expression means they do start producing the protein that needs to be coated around the viral particle. And finally, these viral particles are getting assembled and it needs to go out. So this is what their number is getting increased. So this coronavirus, corona is the name given for the crown. It taken from the sun as the sun rays is going in all direction. The first time the electron microscopy you are seeing here is taken by Almeida and Tyrrell in 1960s, and this lady Almeida was very talented. So this is the schematic diagram of the coronavirus, in which we can see there is a inner coat and outer membrane. This outer membrane will contain 5 different virus contain 5 different protein. In outer area, it contains this spike protein. There are membrane protein, small envelope protein, and nucleocapsid protein. So these viruses have a family called is Coronaviridae. Coronaviridae have 2 subfamilies, Coronavirinae and Tonovirinae. Coronavirinae again can have 3 different genera. It's a general. It's written. It's a genera: alpha, beta, and gamma. So in case of this COVID is name given is that coronavirus disease 2019. So all we know is it spreads through the close contact because of the formats, either through the aerosol droplets or through the droplets or through the touching of the surfaces having the formats of the viruses. So when it enters in our body, in case of the viruses, it utilizes different kind of the surface molecule. So this is one example of the HIV one. This HIV uses this CD4 as a receptor to enter inside the cell, or the cytokine receptor or chemokine receptor. This is called as CCX. In case of this poliovirus, it had a specific poliovirus receptor, or this Coxsackie B or adenovirus has the Coxsackie receptors. In case of the coronavirus, there is a N amino peptidase N, and in case of measles, which has CD46, or in case of rhinovirus, which is a common virus for the cold and flu, is LDL receptor on the membrane. So once it enters in our body, it starts dissolution, assembly, and ultimately these virion particles are released to the surface. Now coming to the origin, we surely not know what is the cause of its origin, but previous experiences is corona-like viruses in 2002 is known as SARS, stands for Severe Acute Respiratory Syndrome Coronavirus 1 or SARS-CoV-1. So this bat is the likely origin source, because this coronavirus, naturally there are 3001 coronavirus species have been identified amongst the living organisms. Out of this 3001 species, only 944 species is known to infect the human. Out of this 944, this COVID, this current one is the most dangerously affecting the human life. So this SARS-1 started origin in the bat, which is believed to be having a intermediate host like wild civet cat, and ultimately it reaches to the human. Similarly, in 2012, same group of virus, coronavirus, outbreak happened in Middle East, and that named as a Middle East Respiratory Syndrome COVID, MERS 2012, and again it had a bat as a common link and camel as an intermediate host and reached to the human. And the recent one we all know is that SARS, Severe Acute Respiratory System COVID-2. COVID-1 was there in 2002, so it's a different strain, so it is called as nCoV-2, and it is also known as coronavirus disease or COVID-19. Again, pangolin could be a intermediate origin, and there is a human infections. Now, this is what this natural source of origin we are proposing, but there are certain glitches in that. What is that? All the coronaviruses are known to have genetic material about 30,000 base pairs. Out of this 30,000 base pairs, they do have 9 major genes which produces 20 proteins necessary for this viral survival and transmission. However, this COVID is having a heavy gene that is around 300,000 base pair. That means 3 lakh base pair. It's a genetic material, and which is very huge. It's not known naturally. So it is still remain to ascertain whether this heavy gene is because of the intermediate host it has passed through and it accumulated this much gene, or it is natural, it is been produced in the laboratory. However, mysteriously the person who has decoded this gene has been missing after 2 days after this publication of this data of 3 lakh base pair gene from the China. Now coming to the symptoms, all we know this common symptom of the COVID is fever, in which that case is very high, that is more than 102 degree Fahrenheit. However, there are in India, there are cases where people do not have any kind of a fever or dry cough, body ache, loose motion. One interesting thing is that it is affecting the central nervous system. Because of the central nervous system, since it goes through the respiratory path in the most cases, so it causes a loss of taste and smell. However, we do not know what is the exact mechanism of damaging this area. CT scan have shown that it can affect the central areas of the brain. So coming to the pathogenesis, so either we do inhale, these viruses enters in our lung. So our lung do 4 or 5 things together. So I'll have your attention in the lower panel of the pictures at your extreme left. You do see this SARS-CoV entry and replication. So it comes inside the body. Since it's a positive strain RNA virus, it replicates and releases there. At the antigen states, what does do? Whenever in the body, any antigen enters our body and affects the cell, and this viral infections, our body destroys that cell. So when it destroys that cell, it helps with the certain antigen presenting mechanism. So it releases the cytokine. So this cytokine includes interleukin beta 1, interleukin 6, interleukin 8, cytokine L2, cytokine ligand 10, 3, 5, etc. So this is called as cytokine storm. Storm means at a small period of time, it releases large amount of cytokines, and there is a problem of this cytokine storm because it weakens the other natural surrounding cells. So in the natural process, the viral infected cells are destroyed by the cytotoxic T cells or CD4 cells. But this virus has a strategy. जैसे कि हमारे वायरस क्लियरेंस में नेचुरली जो इन्फेक्टेड सेल्स होते हैं उन सेल को डिस्ट्रॉय कर देती है बॉडी प्रोग्राम करके, दैट इज कॉल्ड इज एपोप्टोसिस. बट इस वायरस को एक स्पेसिफिक स्ट्रैटेजी है कि CD4 इसको आइडेंटिफाई ना कर पाए और डू नॉट ईट मी सिग्नल देते हैं. जैसे कोई हेल्दी सेल है तो उसमें एक सिग्नल होता है, वो सिग्नल होने से उस सेल को CD8 सेल उसको डिस्ट्रॉय नहीं करता है, एंड दैट सिग्नल इज CD47. दैट मीन्स if any healthy cell which is expressing CD47 will not be eaten by the cytotoxic T cells or CD8 cells. So this signal is called as do not eat me signal. And very cleverly, do not know how this virus stimulates the DNA of the host cell to express more number of CD47. So CD47 means do not eat signal. That means virus can remain inside the cell very easily. Again, the antibody production. So antibody production is okay, but antibody is not being produced if this cytokine storm is too high. That means it's stopping the production of antibodies against it, and it takes the longer time. So coming to the next one is that what is the potential strategies. So first slide I will show you the strategy which was used in 2002 against the SARS-CoV-1, and this red block sign is showing the area where this potential drug were used. So first one is this S protein, you can see here ACE2. ACE is angiotensin convertase enzyme. This is a very common enzyme in the body. So this SARS-CoV-1, which was in 2002, have used this ACE2. So what a strategy people applied at that time to treat them is to stop this attachment with the ACE2. So that's there are certain drugs which can stop here. Again, there is a if this attachment is not stopped, that fusion was stopped. So when this fusion was stopped, it could not go to the next downstream. Again, in the Golgi apparatus, it synthesizes those coating, viral coat. So viral coat synthesis was tried to stop. Again, there is a exit from the cell was also tried to stop. So this is what this vascular damage it causes. Since it goes to the lungs, so this is the normal lung in which we can see there are bronchial epithelial cells, there are alveoli, type 1 alveoli, type 2 alveoli. Type 2 alveoli are having these epithelial cells which creates the mucosa, and there are endothelial cells. So through this, normally oxygen get across. But what happens in the COVID infection? In the COVID infection, because of this cytokine storm, there is a inflammation around this alveoli. You can see there is inflammation, red in color. So it thickens the alveoli membrane. The size of this alveoli membrane is 0.5 micron. So since it's the 0.5 micron, any inflammation enhances the size. So increase in size makes it difficult for the oxygen to diffuse. So hypoxia is one of the condition. Again, this is the in case of the alveoli. In case of the neighboring blood cells, blood vessels, it also damages those blood vessels because of these neutrophil and inflammatory cell attacks. So these blood vessels are getting destroyed. As a result of that, blood is seeping into the lungs and filling it with the mucus and blood. So it making difficult for the breathe. Now, what is the mechanism for that? So this vascular mechanism is being proposed is that there are reacting oxygen species are being produced, and these reactive oxygen species produces this IL beta TNF, and that loses the endothelial junctions. As a result of this endothelial junction removal, what does it do? It enhances the contractility and degrades the whole pathway. Now, coming to the next one. What is the coagulation mechanism? So in the coagulation mechanism, this is the basement membrane, and these are the cytokines, and ultimately these do damage through these receptor, this P-selectin or intercellular cell adhesion molecule, VCAM, ICAM, E-selectin, and this is what this way it to create the inflammation around the lungs, alveoli. So what are the potential target immunologically? So immunologically, there are certain medicines have been produced which are apart from the symptomatic treatment are being given, or the steroids are given to reduce the cytokine storm handling. There are monoclonal antibodies are being also being produced against these TNF alpha receptors and IL-6 receptor. Because IL-6 are one of the cytokines which are responsible for the inflammation. So the drug which are being used, monoclonal antibody, is the tocilizumab, sarilumab, or in case of this IL-6 receptor, it acts through this JAK-STAT pathway. So to stop this JAK-STAT pathway, there is a ruxolitinib or ferrandinib. These are the monoclonal antibodies which selectively stops this JAK-STAT pathway. Or there are receptors through this inhibitory G protein in which we do use this fingolimod, or we can also use this infliximab, adalimumab. These are the monoclonal antibodies to neutralize this TNF alpha. So this virus, we do know, come to know is that this virus is causing a serious damage to the lungs as well as to the central nervous system, and it is escaping the immune system through expressing the CD47 on the infected cells. Infected cell usually reduces the CD47 so that the attacking CD8 cell or cytotoxic T cells can induce them apoptosis signal or eating them. But cleverly, this virus expresses the CD47 on the infected cell so that it creates a niche in which it can probably remain safe. So that's all we do know till now. I would like to thank you all for your time, patience, and listening carefully. Any questions, please?
Thank you very much, sir, for such a nice and informative lecture. I am sure we all must have benefited from today's lecture. We hope to listen such informative lectures in future also. Thank you so much, sir.
Thank you, ma'am.
थैंक यू सर, बहुत अच्छा आपका लेक्चर लगा। मैं चाहूंगा आप अपना जो पीपीटी है वो हमसे शेयर करें तो हम जरा अपने बच्चों के साथ शेयर कर पाएंगे। और बहुत सी ऐसी इन्फॉर्मेशन जो बिल्कुल हमारे सामने से गुजरी नहीं थी